Hello, this is Dr. Divya Pradhan, and today we are going to talk about gene linkage. Well, a single chromosome contains many alleles in definite fixed order. What are alleles? These are nothing but variations of the gene. So in simple terms, in simple language, what we are trying to say is that a chromosome contains many genes, and these genes are found in fixed order. They're not in any random sequence, but their location is very fixed. And this fixed location on the gene, of the gene on the chromosome is called locus or loci. Now, if these alleles or genes are close to each other, they tend to form groups and are inherited together. So, what I'm trying to explain to you is, I'll give you a simple analogy. Two of you and your friend always tend to be together, right? You all tend to go to class together, go out to eat together. So you form groups. Similarly, these genes, they form a group, and these groups are called linkage groups. These genes tend to be inherited together. All right. Now, a good question to ask. Why did Mendel not come across linkage groups? He saw independent assortment, right? Why did he not see this? Well, he did not see this because the two traits that we talked about, that Mendel talked about, for example, tall dwarf and the green flower, as well as the green pod as well as the yellow pod, they were on non-homologous chromosomes. So because they were on non-homologous chromosomes, there was no question of them forming linkage groups. I will explain this further, okay? Now, just let's look at chromosome 19 for an example that's been given in a book. And on the chromosome 19, you can see that there are genes, and these genes have a definite loci or locus, all right, or location. So here, the genes are named, notice, not because of their function, but because of the disease that they may be associated with, all right? Let's see the complete gene linkage. Here is a good example. Now here there are two genes A and B close to each other. Because they're close, they will form linkage groups, and when it comes time for meiosis and crossing over, notice that they will not form any new combinations. No new combinations will be seen. So you will have two gametes, and each gamete will be like the parent, all right? No recombination is seen. On the other hand, if these two genes are far apart, then what you are seeing is, well, you are seeing no new recombination like the parent, but then you are going to see recombinant genes. So you're going to see capital A, lowercase b, uppercase A, lowercase a, uppercase b. So you are seeing two like the parent and two new combinations, which we call recombinant gametes. So here, linkage has not worked. Why has it not worked? Because the genes are farther apart. All right. With this, I hope you understand gene linkage. We will talk about further in other videos. Thank you for your time and patience.